So thanks for the introduction, Manoj. And yeah, it seems like the socializing that's going on at IPC is very impactful. People are really appreciating the time to collaborate and discuss, and I think that's really, really great. I just want to take 1 minute of my time to acknowledge the local Thai team here. Ing, Tar, and Ball. If you see them on the outside at the registration desk, please tell them thank you because they've made my life so much easier. They've done an excellent job at organizing this conference. Yeah, thank you so much. So I'm going to talk a little bit about the work that we're doing in optimizing chili breeding for climate change and the work that we're doing to integrate our genomic approaches for heat-tolerant and high-quality breeding material. So I don't have to do this, but I always like to show how important chili is in Asia. So no matter what color I use or what scale I use to draw this map, almost every country looks the same because China, India, Indonesia are such huge producers of chili. You can see Ethiopia, Nigeria, Mexico, Turkey are a little bit darker and everyone else is almost the same. So Asia really is leading the world in chili production. However, when it comes to yield, it's a very different story. Globally, on average, according to the FAO, we get about 22 tons per hectare yield on average. If we look at Europe, of course, it's much higher. North America is around 32. But then we scroll down to Asia and Africa, we're getting less than half. So if you look at Asia as a whole, it's about 22 as well, but a lot of that is because of East Asia. So China, Korea, these places that have big market segments and also very high productivity. But if you look at South Asia and Southeast Asia, like Thailand, where we are here, yield is very less, so about 12 tons per hectare. Africa is a very similar story with 10 to 12 tons per hectare. And of course, there's a lot of reasons for that. Market segmentation is one, bell peppers compared to chilies, but also stresses, right? Abiotic and biotic stresses. So as Marco mentioned on day one, and that we've talked about several times, World Vegetable Center works with the APSA seed companies, the Asia Pacific Seed Association, and we have a consortium of about 64 seed companies that work closely with us. And every year we do annual surveys and we ask them, what priority traits do you need World Vegetable Center to work on? And this is the latest list that came out. So we have about 64 seed companies, 31 replied to this survey. You can see number one, Bagomo virus. So that's your pepper yellow leaf curl virus, chili leaf curl virus. Number two, anthracnose. Lorena talked about anthracnose this morning. Number three, yield. Surprising. Number four, heat stress. So our users, the people who use the breeding material that we develop, the seed companies, they need our top four priorities, 2 biotic stresses and an abiotic stress. So this really has to be the target of my breeding program. But when we look at heat stress, yeah, it's obviously critically important, but also incredibly challenging. So breeding for heat tolerance is very complex. And it's so complex that I don't consider heat stress tolerance to be a trait. I consider it a phenotype that is composed of many component traits that come together and sum into heat tolerance. Very similar to yield. There's no yield trait. There are QTLs and component traits contributing to yield. So we know that there are several components. Several very weak links in the chain include pollen viability, fruit set, but lots of other traits as well. So I hope you can see in this photo, I don't know in the back, but there's the floor or the ground in this photo is just littered with white flowers. So we went out one day, we had a heat shock, and the flowers just dropped. So it's very common in pepper to see flower drop. We know that a lot of these traits, or almost all of them, are quantitative, so they're controlled by multiple genes. And yeah, so breeding for this phenotype is very complex. So the way we're trying to tackle this is to utilize genomic approaches to effectively and efficiently select for heat stress tolerance component traits and combine them into a heat stress tolerant phenotype. But we also have other challenges from the breeding perspective, not just the phenotype, but from the genetic resources that we have available. So unlike a lot of other crops, particularly tomato, for example, the use of diverse genetic resources, especially wild relatives or land races, is really limited in pepper. Most available molecular markers are also based on biparental populations. The use of GWAS or the core collection, this week or these few days we've had several presentations using the core collection and GWAS, which is a groundbreaking thing in pepper breeding. Most of the populations are biparental, single source, so your marker won't work in my populations. We have a very large genome and very high transposable elements. So 3.5 GB, more or less, 3.3, 3.5, and 80% retroduplication. So the core collection. So we've heard a lot of presentations already, so I don't have to introduce it too much, but it's a huge breakthrough for the breeding program as far as access to genetic diversity. So through the G2P Sol, the EU Horizon 2020 program, we sequenced the entire collections of multiple gene banks and almost 15,000 accessions. And based on that diversity analysis, we selected 423 lines to represent the core. And then we also conducted whole genome resequencing of the core collection. So we have low density sequencing of the total collection and then deep sequencing of the 423 core lines. So the black represents the total diversity within Capsicum, and the orange represents the core collection. So you can see, I think, the core collection is quite representative of the total collection. So we feel quite confident that we were able to capture the diversity that exists and utilize it in the breeding program. So we started this several years ago with partners in the EU and Israel. And we conducted multi-location evaluation primarily for horticultural traits. So we were interested in characterizing fruit traits, yield traits, these types of things. We tested them across 6 environments. So these represent temperate countries, arid environments, as well as tropical environments in Taiwan. And so we made some really good progress in identifying and developing QTLs and SNPs associated with some of these key traits. So the example I give there is the FLEP10, which is a very robust QTL on chromosome 10 associated with fruit length. So you can see here the AA, you get fruit this length, and when you have GG, you have fruit length like this. It's very clear and very robust SNP associated with fruit length. That's great, but it's really, fruit length is not one of the top 5 priorities that we need to work on. So we thought, okay, what can we do to really capitalize on this core collection and really meet the needs of our users? So this is a further example, so some more traits that we worked on. This has all been published in the Plant Journal 2 years ago. But just to show that we worked on a lot of different traits and mapped these so we have environment-specific QTLs or SNPs, and we have traits, location-specific and broad spectrum. So multiple environments, we have robust SNPs. But again, we were interested in heat stress. So what we did is we took this global core collection and we used high-throughput automated phenotyping and the PhenoSpecs, which is a field-based system that collects data every 8 hours throughout the field. So every plant gets evaluated 3 times per day. And we evaluated not all the core collection, but most of the core collection, because that's all that would fit in the PhenoSpecs, across 3 environments. So we have the orange environment is heat stress 1. The blue environment is heat stress, or the control, sorry. And then the red environment is heat stress 2. You can see here, so on I'm having the same problem as, yeah. The left side, which is the right side for me, is the temperature that was measured during the 3 environments. You can see, on average, heat stress 1, the orange, had a higher average temperature during the experiment. But during heat stress 2, we observed a real sharp peak in temperature right at enthesis. So actually, although if you look at averages, it looks like heat stress 1 is more severe, actually heat stress 2 is more severe because of the timing of the heat. So it's not just a matter of exposing the plants to heat, it's when you expose the plants to heat that really plays an important role. But this is just to show you what we observed for the temperature and the solar radiation during the control, which is blue, which is always lower, less solar radiation, less heat compared to the two stress environments. So we took that core collection and we conducted genome-wide association studies across the three environments. And also with Manoj's help, we conducted or developed a STI, a stress tolerance index. And we used the three environmental data to use this STI and also conduct GWAS. So the inner circle is our control, and then the next one is heat stress 1, heat stress 2, and then the last circle, the last ring of SNPs is the STI GWAS. And this is only for yield, but the phenospects, we were able to collect 76 different traits every day throughout the 3 growing seasons. So it's huge data, GBs of data on the phenotypes of these materials. This was great, and we found some really significant SNPs associated with yield traits in the control, in heat stress 1, heat stress 2. But what we're missing is a single SNP or a single QTL across all environments. So we weren't able to find a stable SNP or SNPs contributing to fruit quality traits or yield traits that was consistent across all 3 environments. So we thought, okay, we're able to identify environment-specific SNPs associated with different types of stress, but what we're really interested in is getting durable or, let's say, more robust SNPs. Because we don't want a variety that performs well under stress and poorly under the control. We want a variety that performs well under all environments, right? I think it'll be very difficult to sell a farmer seed. I don't know, I'm not a seed company, but I assume if you told a farmer, under optimal conditions, this variety performs poorly, but under stress environments, this variety does great. I think that's a challenge to convince a farmer. So then we approached genomic selection. So I want to mention, I should have mentioned earlier, and I want to mention now, that all of these genomic selection work was done in collaboration with the University of Hawaii. Anna, who's a postdoc there, and Mikey Kantar, who is our collaborator, helped us do these genomic selection models. So what you have here is you can see the shifts in agronomic genome estimated or genomic estimated breeding values, GEBVs, in the core collection in 13 traits that we identified that had what we call high prediction accuracies. So for us in the core collection, a high prediction accuracy is 0.5. That's been published before to be a threshold as a minimum selection accuracy. For me, it seems quite low, like 50% is the flip of a coin, but they say that 0.5 is a good selection accuracy. And so the dashed line is the medium for each GEB, and what you can see is we found a lot of differences. So again, blue is control, orange is heat stress 1, and red is heat stress 2. So you can see lots of distributions in GEBVs based on the environment. So it kind of validated what we found in the GWAS, and then also provided us a foundation. So what we did is we used this core collection as a training population in our genomic selection models, and then we applied that to the whole collection. Remember, we had this 15,000 accessions across all these gene banks that has low density sequencing. So we used the core collection as a training population and the 15,000 accessions as a test population. And we found there was significant evidence for responsiveness in the capsicum core collection using genomic selection. So what you see here in this figure, this red V is the rank change in genomic estimated breeding values under the 3 environments. So heat stress 1, heat stress 2 on the perimeter, and the control in the middle. So we found these 27 varieties or 27 accessions that when we exposed to heat stress performed well, or they had very high GEBV. So we consider these heat responsive or heat tolerant varieties or accessions in the gene bank. And then we found similar things for cold responsiveness. So at the bottom, this mountain, this blue mountain, these are lines that had high GEBVs under control and lower under the heat stress environments. And then on the right side of the figure, these are the SNPs that were associated with marker effects that were significantly different under the heat treatments for the different varieties. So then we determined the relationship between climate and quality, between the core collection and also this total collection of 15,000 accessions. And we found some very nice associations between the training population and the test population, the core and the global, not just for yield, but also for quality. So we were able to use these models to predict for quality traits under heat stress, as well as yield and other component traits. And again, you can see the red here on the bottom, the red peaks, these are the core collection, and then the blue is the global collection. So you can see differences, but also some very similarities between the core and the global collection. So we think this is a really good foundation to enable us to utilize these new omics tools. I don't know if genomic selection is new anymore, but these new for pepper maybe, these new tools that are available to us to start to capitalize on this huge diversity that exists that's been largely uncharacterized. So we're really excited to continue this work, but the big challenge we find is human capacity. So I'm largely a field-based breeder. I do a lot of my work, or most of my work, is in the field. I don't have a lot of expertise in computational biology or these types of techniques. My team is also learning. We're building our capacity. Manoj is making big advances in his ability to do GWAS and genomic selection, but still we're not quite there. And we think this might be a theme for a lot of breeding programs, not just World Vegetable Center. We feel like it's possible, especially when we look at Asia, Southeast Asia, and Africa, that there's a lot of field-based breeding experience, but when it comes to these genomic techniques, we need some help. So in partnership with the University of Hawaii, we have developed a tool they're called the GEBV Explorer, kind of similar to what Peter was talking about yesterday, that allows you to utilize your data in a drag and drop, easy, simple language tool to do your own genomic selection to help you select parents. And so the real goal is to help the maybe young or upcoming breeders who don't maybe necessarily have the experience or the skills to make very difficult decisions in the field. This tool hopefully can help them be better selectors and able to identify very complex traits in an easy way. So this is not publicly available yet, but it will be. So we have just submitted the publication, so it's under peer review. And I hope that once it's accepted, or if it's accepted, that we can start to publicize it and make it available to everyone. You can use the core collection data, which is also publicly available. You can input your own data, your own traits. It's very user-friendly, and I hope something that you all will use. It's based on LLM, integrated GEBV Explorer. And yeah, I mentioned use a very plain language for breeders, which is very useful for me. This was the big challenge in my breeding program. When a project ends or when a student leaves, and I've heard this from Kevin and others in the discussions we've had, well, the student left, so the project ended, or we didn't quite get to the finish line. So I think this can help me, at least in our breeding program, whether I have a student or not, to be able to do this work. So in conclusion, we are trying our best and hopefully making good progress for all of y'all to operationalize the turbocharging gene bank strategy in globally important crop, which is like chili pepper, that enables the development of both allele mining and population improvement strategies. We're combining genome-wide association studies and genomic prediction using the capsicum core collection, as well as genomic selection on the total collection, not core collection, as a training population. To facilitate the selection of lines for agronomic traits and fruit quality, we created a user-friendly LLM integrated application that was made by incorporating GEBVs for 42 high accuracy traits. And we hope that this approach can enable faster screening of large germplasm collections by rapidly shrinking the decision-making process for parental selection for users even without a strong background in this type of computational biology. So with this, I would like to acknowledge the funders of our program. So, of course, the development of the core collection, the phenotyping of the core collection was all funded through the EU Horizon 2020 program. The PhenoSpecs facility and this work in genomic selection was funded by the Ministry of Agriculture of Taiwan. And of course, we're grateful to the members of the Asian Pacific Seed Association that provide continuous support for our program, not just financial, but also information and knowledge exchange. It's so critical in guiding the breeding program to ensure that we meet the needs of farmers who on the ground, because I think the seed companies really have the best idea of what farmers need. They really have their thumb on the pulse of the farmer needs. So I'm grateful to all that support. Also, this is a bit of an old picture, but still, most of the pepper team is there. There's some new faces, some old faces have gone. But yeah, so of course, I'm here, but this work was by far not done only by me. Huge support from the field team and the collaborators at the University of Hawaii that helped collect the data, manage the trials, and make my life easier. So with that, I'd like to stop, and thank you so much, and take any questions you might have.